Hi everybody, my name is Doug, and today I'll be talking about eutrophication and dead zones. What it is, what causes them, what the effects are, and the methods for preventing them. What is eutrophication? Eutrophication is a process by which a water body becomes enriched in dissolved nutrients that stimulate the growth of aquatic plant life, usually resulting in the depletion of dissolved oxygen. It affects both salt and freshwater habitats. In freshwater ecosystems, it is almost always caused by excess phosphorus. And in coastal waters, the main contributing nutrient is more likely to be nitrogen or nitrogen and phosphorus together, depending on the location and other factors. There are two types of eutrophication: naturally occurring eutrophication and cultural or anthropogenic eutrophication. Natural occurring eutrophication is a very slow process in which nutrients, especially phosphorus compounds and organic matter, accumulate in water bodies. These nutrients derive from the degradation and solution of minerals and rocks, lichens, mosses, and fungi. The second type, cultural eutrophication, which is causing all the damage, is often a much more rapid process in which nutrients are added to a water body from a wide variety of polluting inputs, including untreated or partially treated sewage, industrial wastewater, and fertilizer from farming practices. Causes of cultural eutrophication. Eutrophic events have increased because of a rapid rise in intensive agricultural practices, industrial activities, and population growth. These three processes emit large amounts of nitrogen and phosphorus. These nutrients enter our air, soil, and water. These human activities have emitted nearly twice as much nitrogen and 3 times as much phosphorus as natural emissions. In developed countries, such as the United States and nations in the European Union, heavy use of animal manure and commercial fertilizers and agriculture are the main contributors to eutrophication. Runoff from large agricultural fields enter creeks and bays because of rain and irrigation practices. Fossil fuels and fertilizers also release nitrogen into the atmosphere. This atmospheric nitrogen is then redeposited on land and water through the water cycle with rain and snow. Effects of eutrophication. Now that we know what eutrophication is, what are the effects? Eutrophication can lead to harmful algal blooms, often referred to as HABs or toxic algae. Hypoxia, or the depletion of oxygen and water, eventually, if not treated, causing dead zones. Fish and other water organisms that require oxygen to breathe are suffocated, die, and float to the water surface. In recent years, cultural eutrophication has been invoked as a major cause of seagrass disappearance worldwide. A loss of coral reef and surrounding habitats is also a major effect of eutrophication. Currently, coral reef ecosystems are threatened worldwide by multiple stressors such as changes in water temperature, ocean acidification, increases in salinity in the tropics and subtropics, nutrient loading, and hypoxia. As mentioned earlier, a major effect of eutrophication is what are called dead zones. Dead zones are low oxygen or hypoxic areas in the world's oceans and lakes. Because most organisms need oxygen to live, few organisms can survive in hypoxic conditions. That is why these areas are called dead zones. How are dead zones formed? The diagrams shown compare a healthy water body with a dead zone. As you can see in the top diagram, there is an overabundance of nutrient inputs, also known as nutrient loading, which causes algae to grow at the surface. The algae at the surface traps the bottom body of water from getting enough sunlight needed for photosynthesis. The algae at the top die off eventually and settle to the bottom. The decomposition process of the algae requires oxygen, which it gets from the oxygen present in the water molecules. The fish and other organisms that also require this oxygen eventually die off, causing a dead zone. Another effect of eutrophication are what's called harmful algal blooms, or HABs for short. Harmful algal blooms are a rapid growth of algae or cyanobacteria that cause harm to people, animals, and the local ecosystems. Skin contact exposure from swimming or other activities can cause irritation of skin, eyes, nose, throat, and lungs. Drinking, consuming fish or shellfish, or even breathing in the toxins from the tainted water can cause stomach pains, headaches, neurological symptoms, vomiting, diarrhea, and even liver damage. Harmful algae and cyanobacteria can look like foam, scum, paint, or mats on the surface of the water that can be different colors, as we can see here at the top. How can we detect for eutrophication? There are a few methods that I will discuss in this presentation. The first shown here is what is called a Secchi disc. The Secchi disc is an 8-inch disc with alternating black and white quadrants. It is lowered into the water, usually from a boat or a dock, until it can no longer be seen by the observer. The depth of the disappearance is what is known as the Secchi depth, which is a measure of transparency of the water. Transparency can be affected by the color of the water, algae, and suspended sediments. Transparency decreases as color, suspended sediments, or algal blooms abundance increases. If transparency is measured through the season, from year to year, trends in transparency may be observed. Transparency can serve as an early warning of activities that are happening on the land that are affecting what's going on in the water. A second method to study for eutrophication is satellite imagery. The most useful information for monitoring of eutrophication is water color, as seen in the picture. Chlorophyll pigment and algae is green, contrasted with the blue of the surrounding water. Dedicated sensors like the MERIS, Enviasat's medium resolution imaging spectrometer, and the MODIS, moderate resolution imaging spectrodiameter, can indicate areas which are seeing surface algae growth. A third method for studying eutrophication and early detection of algal blooms is hyperspectral imaging sensors. These sensors can be put on several aerial platforms such as fixed-wing, rotor-wing, drones, airplanes, helicopters, and even low Earth orbiting satellites. Hyperspectral imaging, HSI, is a technique that analyzes a wide spectrum of light. Instead of just assigning primary colors red, green, and blue to each pixel, the light striking each pixel is broken down into many different spectral bands in order to provide more information on what is imaged. Early detection of microscopic levels of chlorophyll allows scientists to better prepare the public for potential devastating harmful algal blooms. Along with detecting and monitoring for eutrophication, there are several methods for prevention. Methods for prevention and reversal of eutrophication include minimizing point source location pollution, the U.S. Environmental Protection Agency, EPA, defines point source pollution as any single identifiable source of pollution from which pollutants are discharged, such as a pipe, ditch, ships, factory smokestacks, etc. A second way for the prevention of eutrophication is minimizing nutrient pollution from agriculture and other non-point pollution sources. A third way is restoring riparian zones. Riparian zones are strips of land adjacent to water that are covered in foliage that act as a buffer between the water and excess nutrients. A final way, that's more controversial, are the installation of bubblers or aerators. Much like a fish tank, bubblers provide oxygen into the water while stirring up the water so algae cannot form. Finally, I would like to share with you a few resources that I found of good use covering eutrophication. The USGS has conducted numerous scientific projects relating to hypoxia, nutrient loading, algal blooms from all over the United States. River valleys, lakes, estuaries, palustre, and freshwater habitats. It's very interesting. I've included the link here for you. Beach combers is another useful resource for people that are living in California that might be interested in being a member of their citizen science project. Their objective is to obtain information on rates of stranding for all species and animals in Monterey Bay, California. The long-term objectives of the program are to provide a baseline of information on average presence of beach-cast marine organisms to assist the sanctuary, the early detection of mortality events triggered by natural and anthropogenic environmental agitation such as harmful algal blooms, hypoxia, and oil spills. The EPA, finally, also has a wealth of information including research papers, scientific data, findings from around the country, and some even around the world. Finally, I have included a story map if you're interested. Reach out to me. A story map that I've conducted for another class that covers the same topic using NOAA data, discharge point locations, as a method to monitor and analyze harmful algal blooms. Thanks for taking the time to listen to my presentation. I look forward to any discussions you may have in the next couple of days. Thank you.